This morning I'd like to go over exam 2, and I'll keep these notes up throughout the remainder of the semester, and I hope it'll be a useful study tool for you, along with the notes that are available. The grades ranged on this exam from a 92 down to a 25. The letter grades, there was a wide distribution of grades there also. So as we go through the questions and answers, jot down any questions that remain in your mind, and if you can't find them in the individual PowerPoint presentations, be sure to get with me here in the next week or so. The final exam will be comprehensive, so this material, you'll see it again. In some instances, I'll use the same question but change the answer choices, or in some instances, the answer becomes the question becomes the answer. So try to pay attention to what's going on here as we move through the multiple choice questions and short answer questions, and really become familiar with the topics or the techniques, the concepts, so that you're comfortable with that going into the final exam. As you're aware, the grades are posted, and then there's some other points that have been posted on Blackboard also in 2 separate columns there. Some more discussion about the lab, and that'll be coming this afternoon with some additional photographs and notes in your lab section at either 1 p.m. or 3 p.m. So let's move on to the individual questions and answers, and we'll begin with number 1. When in the early stages of adventitious root formation, differentiated cells are triggered to form new meristems, it is called dedifferentiation. This is suggesting that a cutting to form a new organ, roots, they're specialized cells, differentiated cells that must form a new meristem, new parenchyma cells that are capable of dividing and reorganizing and giving rise to that new root organ. So this is dedifferentiation. Number 2, the location of root initial formation in monocots and herbaceous plants is in or very near the vascular bundles. I would suggest, if this doesn't make sense to you, to maybe go back and review stem anatomy of monocots. Vascular bundles are scattered throughout the stem, and that should answer your question. If not, let me know. Different stem organization in woody dicots, and a little bit different location for root initial formation in woody dicots in number 3. So here, it tends to be in or very near cambial tissue. Again, a review of stem anatomy will be helpful in understanding that response. So now let's go on to number 4. Most plant species have the ability to form root initials in stem cuttings. However, the presence of a sclerenchyma ring may prevent them from penetrating the epidermis and subsequent adventitious root formation. So this is a layer of sclerenchyma cells beneath the epidermis that becomes almost a barrier-like that can slow down or inhibit roots from breaking through that stem out into the surrounding soil. In our presentation on the principles of root formation, go back and look at some of the slides of individual stems that had been magnified, and we denoted some of that sclerenchyma tissue, especially in some of the more mature, woodier plant tissue. So on number 5, by hedging back stock plants, or sometimes through tissue culture, mature phase material can revert to an easier to root juvenile or transitional phase. We call this rejuvenation. Rejuvenation. So again, thinking about the physiological age of a plant, a more mature stock plant, rooting percentages go down. In other words, the cuttings that are taken from those older, more mature plants don't root as easily. We can rejuvenate that plant by returning it to this more juvenile state of growth. In number 6, girdling is a technique of wounding a potential stem cutting on a stock plant whereby the phloem is disrupted and the rooting potential is increased. Perhaps in your mind's eye, think of a plant, and a single stem on that plant is going to serve as a propagule, a cutting. But before we cut it off of the mother plant, we're going to girdle. Maybe in your mind's eye, see a twist tie around that stem, and it's on there not so tight that it's going to cut it, but tight enough to restrict the downward flow of sugars through the phloem system. And they accumulate above the point of girdling. Going to number 7, the two most common treatments to relieve seed dormancy, that would be scarification and stratification. Number 8, both leaf and root cuttings must form adventitious buds and adventitious roots. This is suggesting if I take a single leaf or a piece of root, both of them have to regenerate not one but two new organs. Adventitious roots and adventitious buds. Those buds, of course, grow into the shoot system, the above ground portion of the plant. Number 9, a question about seed dormancy. And photodormancy describes the dormancy condition where light is required for germination of the seed. Okay, 10, preformed root initials is the answer. They generally lie dormant on the stem until the stems are made into cuttings and those root primordia emerge as new adventitious roots. In lecture, we talked about a few plants that exhibit this: coleus, hydrangeas, some of the vining plants, and I had some photographs of those preformed root initials. Number 11, cuttings taken from stems of dormant plants and rooted in media are hardwood. What's key there is dormant plants. Number 12, proper polarity of stem cuttings in plant propagation would suggest placing the proximal end of the stem into the media for root formation. 13. So this goes to our discussion on hardwood cuttings, and there were 4 production systems, 4 methods of growing hardwood cuttings. This one, dormant hardwood stem cuttings taken, treated with auxins, placed into an environment that's warm, 65 to 70, for a few weeks. This is best described as warm temperature pre-treated hardwood cuttings. So again, that's one of the 4 systems of producing hardwood cuttings that we described in our class. Going to 14, dormant hardwood stem cuttings taken and treated with auxins and placed directly into the soil in the field are best described as late fall hardwood cuttings. That was another one of those 4 methods of production of hardwood stem cuttings. Number 15, etiolation is a localized light exclusion pre-treatment of stock plants which excludes light from a portion of the stem. 16, auxin. On this one, there was just, your choices were several plant hormones, and auxin is the correct answer for promoting adventitious root formation. 17, cuttings stored for a few hours to a few days should remain in cool, moist conditions until placed in the propagation environment. In other words, you receive some cuttings. You're not ready to stick them in soil. Can't just let them sit in your office. or out in the greenhouse, best to get them into a cool, moist environment. Going on here, rejuvenation of stock plants. Again, this was part of our discussion of stock plants that were in a mature phase and how can we return them to a more juvenile form so that the rooting percentage is increased. And we talked about the use of cytokinin sprays and gibberellic acid sprays, whereby that has shown some success in some plant species of rejuvenating them. Going on to our next two, 19 and 20, it's a description of some of the grafting techniques we went over right before the exam. And the first one here on number 19 is a whip and tongue graft. That is a description of the whip and tongue graft. And 20, this is a top working technique, and it's describing cleft grafting. So if you have questions about 19 or 20, go to the techniques of grafting PowerPoint, and these are described, and there's a couple of videos to watch on each one also. In 21, in tree grafting, the benefits of certain rootstocks include, you had four choices there, and the correct choice was D, all of the above. So the previous three were greater resistance to stress and disease, controlling size, and hastening reproductive maturity. Going on to 22, the naturally occurring plant auxin that was first isolated and characterized, that was IAA. Indole acetic acid. So that wraps up the multiple choice. Then we move on to some short answer. The first short answer is list and describe the three phases of seed germination. So again, seed germination. You place the seed in soil, add water. How do we describe germination? We can do that in three phases: imbibition of water, where it swells. The second phase is a lag phase, where the seed appears to be sitting in the soil, but actually there's a tremendous amount of metabolic activity occurring in this lag phase. It just doesn't increase in weight or length. And then finally, radical protrusion. So here the length begins to increase as the primary root, the radical, emerges into the surrounding soil. And this describes the phases of seed germination. Next short answer. As a propagator, what would be the advantage of clonal rootstocks over seedling rootstocks? Here, go back to our discussion of the principles of grafting and budding, whereby the grower needs to make a decision about seedling rootstocks or clonal rootstocks. And we made some comparisons between the two. The advantages of clonal rootstocks over seedlings is primarily that of uniformity. The clonal rootstocks, they tend to be much more uniform in their phenotypic characteristics. Maybe to a lesser degree, you might prefer clonal rootstocks because it allows you to influence the final size and growth habit of the grafted plant. It might, if you choose the proper clonal rootstock, allow you to have a dwarf or super dwarf fruit tree. Moving to the next short answer. What is KIBA and what is the significance of K? This is potassium, K being potassium, IBA, a rooting hormone. When IBA is manufactured, there's a step where they can add a potassium ion, makes the molecule polar, and thereby allows it to be dissolved in water. The KIBA can be dissolved in water. IBA alone cannot be dissolved in water. Our next question is about correlative effects. This is a concept that we discussed in our section on root formation of cuttings. And it's a concept that evaluates or describes how one plant organ has control over the growth and development, maybe, of another plant organ. Maybe think of it as a leaf has control over root formation, or maybe a bud has control over root formation. So this is the idea of correlative effects, and it's mediated primarily by hormones. Going on now to a question that was taken from your section on the propagation environment. And in a couple of slides, we talked about organic and inorganic components that we combine together to make a soil, a media for propagation. Inorganic components are those that are listed. And again, I'm asking you to present 4 of them and describe them. There are potentially others that are more minor, and depending on which ones you listed, I did accept some of those. All right, going on now to the final question. Coming back to grafting. In our concepts of grafting, we talked about the importance of lining up the cambial tissue, vascular bridge formation, but there was one slide specifically, and I've essentially copy-pasted that slide onto this one, describing those genetic limitations, and that the further apart the plant species are, the cultivars are from one another genetically, the less likely they are to take. The less likely they are to heal and form one composite plant. So in other words, if it's clones, Sam Houston peach onto Sam Houston peach, no problem, should be 100% success. If I'm grafting 2 different clones onto each other, same genus species, I should still have a very, very high percentage of success. And the example I gave you there was grafting a Sam Houston peach onto a June Gold peach. Then you take a step back. What if it's the same genus, but different species? So what if you have Prunus armeniaca and Prunus persica? This is grafting apricot onto peach, which is acceptable. You've taken a step down in terms of success. It might be closer to 70 or 80% chance of success if they're 2 different species, same genus. But, okay, so that would be the same genus, different species. If you have 2 different genuses but the same family, here the example, Citrus sinensis is orange, and you're grafting that onto sour orange rootstock, which is a fairly common occurrence in citrus grafting. It is successful. Nonetheless, this isn't commonly successful. We might say 20, maybe 25% successful as we get this far apart genetically. Then in the same family, different families, wow, this is nearly impossible. There aren't any documented cases of taking a plant from one family and grafting it onto a plant in a different family. So again, to summarize, the further apart they are genetically, the less likely they are to heal properly and form a successful graft union formation. So this takes us up to the last question of the exam. Again, as you review through the notes here, and again, I'll leave it up for the remainder of the semester on Blackboard. Let me know if any of these need clarification. And again, lab will be coming at you this afternoon